scientific historical fact number 2. We're told that these viruses are found, discovered. That's not at all what they do. The steps that virologists do is they take a sample from a human being who's sick. They centrifuge it, filter it, then they put it on a cell culture of monkey kidney cells with antibiotics, bovine calf serum, other substances. The point is that it's a mixture of stuff. They didn't separate out this tiny particle all by itself. They reduce the nutrients which feeds the culture breaks down, it decays. And they claim that the decay, the dying of the culture is proof there's a virus in there. The term they came up with to describe this is called CPE, cytopathic effect. And the problem with this is that when they do a scientific control, which means they do all the same steps without a human sample, they get the same results, which basically disproves the whole thing. And even the guy who came up with it, Enders, he said, when we didn't add the heavy metals and did the staining and all this, we got the same results. And another microbiologist, Stefan Lanka, has done controls with just yeast and with other materials, and he got the same results. That method has proven flawed, yet they still use it. And so that's why they try to steer us towards the more technical stuff, which is like the PCR test, antigen test, genomics, or sequencing. The problem with all of these is they assume there's a virus in there to begin with. So there's no gold standard. The other problem with all these, they don't do controls. By that I mean, you send these labs 5 samples. Let's say one's lung cancer patient, one person has pneumonia, one person is declared to have COVID, somebody else is declared to have the flu, somebody else is said to have a cold. You blindly label these vials. You send it off to the lab and you say, okay, tell us which one's which. And they should be able to do that. And then you repeat it. You just send it to other labs with different samples, and they should be able to tell you which one's which. That's science. That's how you prove that this thing is its own entity.